So my name is Åsa Johansson, and I'm one of the scientific leads for the precision medicine capability. And I will today present to you this clinical technology developmental project that we have recently launched the call for. So the agenda for the meeting today is I will give a short presentation of the call and a little bit information about SciLifeLab. That will be followed by some questions. You can write questions in the Q&A during the speak, or you can raise your hand and ask them directly after the presentation. After this question, we will follow by short presentations by the SciLifeLab platforms, about 2 minutes per platform, followed by questions in different breakout rooms. You can choose to go to the platform that you are interested in discussing further with. So for you who doesn't really know SciLifeLab very well, we're just going to give a short introduction to SciLifeLab and the capabilities. So SciLifeLab comprises of 3 different dimensions or pillars, which is the research community, the data-driven life science, and the research infrastructure. The research environments comprise 190 affiliated research groups with more than 400 SciLifeLab researchers, representing a broad field of science, including mainly basic science and to a less extent clinical research. The infrastructure consists of 10 technology platforms organized in 40 research units or infrastructure units with more than 1,400 users yearly and 3,000 projects per year. And about 500 technology experts are employed. The most recent initiative is the data-driven life science that was initiated to accelerating a paradigmic shift towards data-driven life science with 3 strategic research areas, which is one of them, the precision medicine and diagnostics, which might be the one that is most related to the precision medicine, but also the epidemiology, infection biology that is very related to the PLP program. So about 2 years ago, or a bit earlier for the PLP, this capability idea was launched at SciLifeLab with the idea to try to create a cross-platform and cross nodes and SciLab site collaborations or initiatives where we focus on different research area. And first out was the pandemic laboratory preparedness that was initiated already during the COVID pandemic. And after that, the precision medicine capability was launched 2 years ago. And the call today is a collaboration then between the precision medicine capability where me and Eva Berglund is here today, and the pandemic laboratory preparedness, PLP, where we have Staffan and Alice here today. So the clinical technology developmental project is then an initiative to try to strengthen the collaboration between the SciLifeLab infrastructure and clinical research in healthcare. As mentioned, we are doing a lot of basic research today and provide services for basic researchers, and we would like to strengthen the support that SciLifeLab gives to clinical researchers in healthcare. So the idea with the clinical technology developmental project is that we could, for example, adapt new technologies for well-defined precision medicine applications. It's supposed to lead to an application that has a potential to advance diagnostics, treatments, or follow-up of patients. And there should be some specified timeline for clinical implementation of the project. We haven't decided upon a specific number of years because this will depend a lot depending on what project you're working with. So who can apply for this funding? Each proposal is expected to have at least 2 applicants that links the SciLifeLab infrastructure or the PLP unit, who are supposed to be the main applicants, to a clinical researcher, a hospital clinic or a clinical laboratory, who will act as the co-applicant here. The SciLifeLab infrastructure unit should be represented by the head of the unit, but we can have other affiliated scientists as co-applicants as well. The PLP units are defined as grant recipients of the earlier PLP calls, the PLP1, PLP2, PLP clinical micro, and PLP test. Each unit will be able to submit one application as main applicant. And we are open to discuss with the larger units whether they can submit more, because the unit definition at the SciLifeLab is not identical across the different platforms. So, for example, NGI and NBIS, which are huge, very big units, we might be able to discuss whether you can submit more applications. And each individual can only submit one application, meaning that you cannot submit both for PLP project and for a precision medicine project. So the total budget for the call is 4.2 million crowns. And of those, 3.2 is directed to the precision medicine capability project, whereas 1 million is for the pandemic laboratory preparedness. The typical grant size will be around 300 to 500,000 crowns, and the funding will be allocated to the SciLifeLab infrastructure or PLP unit to cover reagents, materials, salaries, or other running costs. And this is also why we would like to have the head of unit as an applicant for this, because that is the person who will be responsible for the funding. So we are not able to give any funding to research time for clinicians, unfortunately. So what can you apply for? The idea is to support well-delineated projects addressing an unmet clinical need, where technology development in collaboration with one or more SciLifeLab units or PLP units could enable clinical applications. The technology that the clinical application would be based on can be established already at one of the units, but it needs to be some additional development or validation to enable it for clinical use. So relevant samples for the project should already be available so the project can start quickly, and ethical permits should be in place if such permits are needed. So just some example what a clinical TDP could be is, for example, assay development or validation, optimization with respect to minimizing sample requirements or reducing turnaround time, developing quality metrics, for example, to ensure absolute quantification, establish streamlined pipelines for analyzing clinical samples, also to some degree including data analysis and or clinical decision support. And some examples what we do not support with this application is basic discovery research that involves using existing technologies for just analyzing new samples. We don't support identification of new biomarkers from scratch, for example. We don't support development of completely new technologies because that's a bit out of the scope of this call. We don't support projects aiming at therapeutic innovations and not projects only focusing on data analysis and or clinical decision supports with already available data. So there need to be some analysis, molecular analysis as well. We can also offer bioinformatics support to some project up to 100 hours after feasibility evaluation by NBIS. And if more extensive bioinformatics support is needed, NBIS should instead be considered as a co-applicant. So please contact NBIS if you're interested in that and discuss the possibilities. So the evaluation process, all applications that fulfill eligibility criteria will be evaluated by an external review committee consisting of experts within the precision medicine field. The assessment will be done using the following criteria: clinical significance, scientific quality and feasibility, and added value of the grant. The funding will support a diversity of projects with regards to, for example, gender and the applicant, career stage, university affiliation, technologies used, and disease areas. And the timeline, the call opened a few weeks ago, and we have the information meeting today. The deadlines is April 10th, and the plan is to have a quite quick review to be able to have a final decision of which project will be funded at the SciLifeLab board on May 28th. And the decision will be made public in June. The project is supposed to start on September 1st and go on for 1 year. So with that, I'm happy to take any questions that you have so far before we leave the words to the platforms. I don't see any questions in the Q&A. And I don't see anyone raising your hand either. Okay, I think that we move on then to the platform presentations. So we have 10 platforms at SciLifeLab, and I think if I count correctly, that we have 9 presentations here today. We will start with the genomics platform with Tuuli Lappalainen, if you're here.
Yes, I'm here. Hi, everyone.
Okay. And just tell me when I should change slide for you.
Yeah, I think you can do it anytime. Yes, so hi, everyone. My name is Tuuli Lappalainen. I'm the platform director of the genomics platform, and also the director of the national genomics infrastructure, which is basically the vast majority of the platform consists of NGI. So in our platform, we provide expertise, infrastructure, equipment, services in everything having to do with sequencing technologies and genomics. We have a very comprehensive technology platform, including all the major sequencing technologies and other omics technologies to really analyze things ranging from different genome sequencing approaches to epigenomics, transcriptomics, other functional omics readouts, also proteomics and others from very different types of source materials, ranging from tissues and cells to, of course, nucleic acids, different types of biospecimens. And these are applied to basically the entire scope of biomolecular science, and especially in the sort of infectious disease and clinical space, we do a lot of work here. Of course, we don't really do. Malin will talk more about the clinical genomics collaboration and how we sort of integrate our activities. We don't do so much of direct analysis of samples for clinical decision making, but we do a lot of technology development and assay development, thinking about how we use these technologies and capabilities that we have to develop new ways of using omics in health-related research and clinical decision making. So yeah, we will be hosting a breakout room as well, where you can ask more questions.
Okay, thank you very much, Tuuli. So next up is clinical genomics and Malin Melin. See if you are here.
Yes,
Yes.
I am.
Welcome.
Yep. So now we're switching to clinical genomics, where we work in close connection to healthcare, and our aim is to improve diagnostics and facilitate precision medicine by supporting translational and clinical research. And we don't really perform any discovery research. We leave that to NGI and the genomics platform, but we work in this translational and clinical research field, where we try to form a bridge into healthcare to accelerate the use of new genomic technologies. And we set up and adapt methods for diagnostics in different research projects and also for healthcare. We collaborate, as Tuuli mentioned, on the method development as well with NGI. So our key technologies are human and microbial sequencing using NGS, but also other technologies such as digital PCR, for instance, to monitor cancer patients during treatment. We also have, for instance, single cell technologies. And we provide tailored support, adjusted for what is needed in the different projects, and that includes clinically validated tests, but we also set up new tests, for instance, gene panels or whatever is needed in the different projects. And we're used to challenging samples, real clinical samples, and we perform both retrospective but also prospective studies that may need a very rapid turnaround time. And we have in-house expertise on clinical diagnostic that can be utilized in the different projects. We work in 4 different disease areas: rare diseases, solid tumors, hematology, and clinical microbiology. And the wet lab parts are combined with bioinformatics, informatics, and we also can provide clinical interpretation of the results if that's needed, and even clinical reports, for instance, for clinical trials. So that was a very quick overview. I'm happy to answer questions in the breakout rooms later.
Thank you very much, Malin. So next speaker from the metabolomics platform is Otto Savolainen.
Hey everyone, you can just switch to the next slide. Yeah, my name is Otto Savolainen. I'm presenting the metabolomics platform. Where we have Yeah, I will continue. There are some mics open, I think. But we have the mission to providing expertise and service in small molecule analysis, and especially focusing on metabolomics and exposomics. And our platform has 3 different nodes in Sweden. So we have our largest node, the Swedish Metabolomics Center up in Umeå, where there's a collaboration between SLU and Umeå University. And then we have our exposomic facility in Stockholm University, and then Chalmers mass spectrometry infrastructure in Gothenburg. Our 3 nodes are focusing on mass spectrometric metabolomics and exposomics, but then also at the wider SciLifeLab umbrella, there's also both spatial and NMR-based metabolomic services available, and those are represented by other platforms today in this meeting. You can jump to the next slide. Can you? Yeah. Our platform provides access to both targeted assays and to untargeted screening services. And on the targeted side, we have a wide range of different targeted assays available, tailored for different matrices. And then on the untargeted screening services, all 3 different nodes provide untargeted screening services, but there's a bit of a special niche in each node when it comes to the untargeted part. We have also just engaged recently as a platform to development and implementation of a very wide range of metabolomics or targeted quantitative metabolomics panels at the platform that we foresee will cater to the precision health need in a good way by being able to provide easily accessible phenotypic data into studies and into different activities in this field. We focus on both endogenous and exogenous metabolites, and then we also have a special focus on chemical exposomics in the exposomics facility in Stockholm. Then also from the past and also recent focus is also on health-related exposures that we provide as a service at the platform. And overall, our focus has been quite strongly and is strongly on precision medicine and precision health. And at our platform, we have a number of development activities that we have identified that we believe will cater to the need of precision health in Sweden. And with these activities, we are aiming at enabling metabolomics as one of the key techniques in this field in the future. Thank you.
Yeah, thank you very much, Otto. So the next platform is the cellular and molecular imaging presented by Rebecca Howard. Welcome.
Hello, you can hear me, I hope. Yes, thank you. So the cellular and molecular imaging platform enables research that requires imaging on all kinds of spatial scales. So that's from the atomistic to organismal, both by leveraging and also integrating really advanced fluorescence and electron microscopy methods. So technologies that may not be generally accessible to individual research groups or local facilities. And in terms of the vision for the future, CMI is really aiming to strategically sustain and also expand our imaging methods, and especially develop multimodal approaches that are kind of at the interface between imaging applications. And obviously also to spread these techniques to a wider range of research fields, which would certainly include medical science. Specific components of the platform are organized into this integrated microscopy technologies unit that's shown on the left here, which includes in green the advanced light microscopy node in Solna, our kind of core national facility, which has a wide range of super resolution fluorescence and single molecule methods, including MinFlux, nanoscopy. In orange, also the so-called FibSem method, which uses sample milling to reconstruct volume EM images for example, cellular substructures, and that's up in Umeå. And in blue, this really developing correlative array tomography method, which integrates fluorescence and high-resolution EM imaging, and that's based in Gothenburg. On the right, we also have our electron cryomicroscopy or cryo-EM unit, which has nodes in both Solna and Umeå with Titan Cryos, as well as screening microscopes at both locations to assist with both cryo-electron microscopy structure determination, typically of macromolecules, and also increasingly cryo-electron tomography imaging and microelectron diffraction methods. And those are also supported by an integrated network, which is shown in blue there, with screening sites distributed around the country, including in Gothenburg and Lund and Uppsala.
Okay, thank you very much. So next platform is the spatial biology presented by Charlotte Stadler.
All right, thank you. You can go to the next slide then. Yes, so the spatial biology at SciLifeLab, our aim is to provide service for advanced spatial analysis of biomolecules in situ. So we work with tissue sections that can then be patient material in terms of precision medicine focus. And we have various methods where we analyze different types of omics. So it could be genes, transcripts, proteins, peptides, or metabolites. And what you see at the bottom is just a few examples of data that's been generated within the platform, ranging from global gene expression with Visium down to sort of neurotransmitter expression with MALDI imaging. And what I think might be the most interesting in this particular forum is the ability to do multiplexed immunofluorescence to look at many protein markers in one tissue sections as a way to modernize image-based pathology. And I thought I'd just give two brief examples. So if you go to the next, this is one project that we're currently working on that aims to move away from single-plex immunohistochemistry to a 10-plex multiplex panel in order to accelerate lung cancer diagnostics. So we can take all of the clinically relevant markers that are today used in cineplex IHC, and then we make a panel with all of these 10, and we then make use of one single tissue section in order to sort of better profile the different types of, in this case, lung cancer tumors. So it's a way of sort of modernizing diagnostics in cancer. Another example, if you go to the next slide, is that this could also be done sort of more to improve diagnostics and treatment selections in the future. So this is a bit more of a discovery approach where, considering that we can look at all of these different protein markers together, we can use them to define cell types, cell distributions, and expression of new therapeutic targets, all in this very same sample. And the aim of these types of projects would then be to link the spatial profiles and the tumor profiles to clinical outcomes if we look on retrospective cohorts that can then be taken to analyze more prospective samples to guide in treatment selection. And this is just one example of an ongoing project where we look at expression of therapeutic drug targets for new treatments in bladder cancer. So if you have questions, you can see the email address, and I don't know if I will have a breakout room, but I will stay as long as the meeting is if anyone would have questions. Thank you.
Yeah, I think there will be a breakout room for you as well.
Okay.
So the next platform is the chemical biology and genome engineering presented by Anna-Lena Gustafsson.
Actually presented by me. My name is Bernhard Schmierer. I'm the co-director and coordinator of the platform. And can I get the next slide, please?
Yes.
So our platform is a multidisciplinary platform. We have 3 units. The biggest one is Chemical Biology Consortium Sweden. So the Chemical Biology Consortium Sweden, or CBCS, they handle everything that has to do with small compounds. So they do chemical screens, they do assay development, they do disease and compound profiling. The second unit is chemical proteomics. They use proteomic techniques to study the reaction of cells or which proteins bind to small compounds that one exposes a cell to. They have different methodologies available here. And the third unit is CRISPR functional genomics, focusing on one hand on pooled CRISPR screens, so genetic screens that can be genome-wide or custom screens, but also on precision genome editing for model system generation. Next slide, please. So we apply orthogonal approaches, genetic, chemical, and proteomics, and all together we can use this for mechanism of action elucidation of small compounds, mainly as a platform, but we're of course not limited to this. We can also do studies in functional biology just by generic gene perturbation or chemical perturbation, and we have relevance to precision medicine, especially in looking at for example, isogenic cell line models that can be made by CRISPR. Can I get the next slide, please? So this is we do a lot of things. So here I just outlined here what we can do for you. So if you want to, for example, identify, study, or improve small molecules to affect any biological process or phenotype of interest, then Chemical Biology Consortium is your go-to place. If you want to identify proteins or genes that are involved in a cellular response to a small molecule, that we can help you with this in the platform. If you want to identify genes or genetic elements possibly that regulate certain biological processes, so you can discover new biology in an unbiased manner, for example, by CRISPR screening. And maybe most relevant to this audience here, if you want to generate disease-relevant models by precision gene editing, and then screen this for drugs, for example, or do whatever you like with them, so make proper reporter systems, create isogenic cell line pairs, then this is also something we can help you with. I have also a breakout room afterwards, I think. So thanks for your attention, and I'll take questions later.
Thank you very much. And next platform up is drug discovery and development.
Yes, thank you very much. My name is Paweł Baranczewski, and I'm head of ADMA of Therapeutics unit within drug discovery and development platform. Next slide, please. And we can actually do, in terms of clinical science and precision medicine, different things like in vitro administration, distribution, metabolism, excretion profiling, bioanalysis, and determination of pharmacokinetics profiles, also bioanalysis and monitoring of different biomarkers, and more specific study like determination risk for drug-drug interaction, metabolite identification, mechanistic study, and try to investigate population variability. Next slide. And with all this information and experiments using mechanistic physiological-based pharmacokinetic simulation and the different modeling techniques like pharmacokinetics, pharmacodynamics, and population PK modeling, we can actually understand drug disposition in patient body, estimate exposure and intra-individual variability, also correlation between exposure and effect, understanding the reasons for inter-individual population variability, identify covariants contributing to the population variability, and on the end, of course, evaluate effect of intrinsic and extrinsic factors which may affect the drug in our body. Next slide. We can also, as platform, we have, of course, access to state-of-the-art laboratories, and we have competence to support design in vitro and in vivo pharmacological studies. And together with the preclinical cancer treatment center, we can provide animal studies on pharmacokinetics and pharmacodynamics. Next slide, the last one. So as platform, we are special place because we have collaboration with innovation center, with academics group. So we have and scientific edge, we have business development expertise and know-how how to make treatment for the patient which is needed. We work with different kinds of molecules, small molecules, antibodies, oligonucleotides, and we are over 50 with industrial, most of us with industrial, pharma industry background, and have also access, of course, to academic research. And as contact, it's Rebecca and myself. Thank you very much for your attention.
Thank you very much. And I think that we don't have Claudia with us here yet in the meeting. So we move on to the bioinformatic platform with Björn Nystedt.
Yes, hello. So I'm Björn Nystedt. I'm the co-director of the bioinformatics platform at SciLifeLab, if I get the next slide. So the bioinformatics platform work, one of the major things we do is to work very closely with the research groups to work at custom tailored bioinformatics analysis of all sorts of life science data. So if you're thinking about projects where you think that where you need to work out the best way to analyze or visualize your data or to establish new workflows for the data processing, then it's probably a good idea to contact us in this call. And as also said, we have set aside some budget already for this call as part of the call to help all of the ranked projects to some extent. But if there is a bigger bioinformatics component, then we should be the applicant or co-applicant of those projects. And in principle, we have 3 units in this call that are relevant, and one is more classic bioinformatics, if I put it like that, working with all sorts of omics data, genomics, proteomics, whatnot, including pipeline development, but also more AI and sort of machine learning parts. And then we have one unit that is very strong in bioimage analysis, both more classic bioimage analysis, but also a lot of the modern deep learning methods and so on. And so they can handle all sorts of bioimaging, sort of data analysis and data processing. And then we have a unit called the IDA Data Hub, which is in a way the most closely integrated with the clinic right now. And they work with medical AI imaging data. So if in case you have digital pathology data, for example, this is a good partner for this call. Yes, so these are the units, and I put up some of the competence areas to the right there that we cover. I should also stress that there are also, of course, relevant competence for data analysis at all the other platforms, and we and the other platforms are also very happy to collaborate across platforms. So don't hesitate to contact more than one unit or platform. And if you don't know where to go, we will also help you to connect between the different platforms if that is needed. So I just wanted to complement that also from what you said in the beginning.
Yeah, absolutely. Thanks a lot, Björn. And now I also know that Claudia is here. So we take the last platform then, which is the clinical proteomics and immunology.
Yes, thank you so much. And sorry if I couldn't join the full meeting. So the clinical proteomics and immunology platform, I think that it's quite relevant for this call in general. So our aim is to advance discovery, personalized medicine, and providing cutting-edge technology and cutting-edge technologies in proteomics specifically. So we have expertise, infrastructure, and instrumentation to run different types of proteomic experiments. So from affinity proteomic, one very popular method that you may have heard of, it's Olink and Olink Explorer. So the profiling of an high number of proteins using antibodies. But we also have the possibility to apply high density array, high resolution mass spectrometry. And using this different type of technology, we are capable of analyzing human samples, and the units in Stockholm are equipped with certificated as BCL2 labs. So our services or our expertise is in general in biomarker discovery, both in biofluid, but also in tissues and cell samples. And we are capable of doing molecular phenotyping, and we have been involved in projects of drug development. One of our big achievements the past year, since we have the high density arrays, was to support the serology analysis during COVID. So we have analyzed more than 300,000 samples, showing our capacity and to provide services for large project with healthcare as well. And in our case, we have one of the units, which is the affinity proteomic unit, which I had specifically, is involved already in one of the pandemic laboratory preparedness. And what we are doing, actually 2 units. Affinity Proteomic is working with a pandemic laboratory preparedness project focusing on microsampling, a new sampling type that can be used to monitor biomarkers in the population, for example. And then we also have the autoimmunity and serology profiling, which actually is involved in the pandemic laboratory preparedness as well, for their capacity to profile antibodies against different types of infectious disease. If you go to the next slide, I would say that more in general, we have a great capacity to support projects or clinical projects. Our strength is that having both capacity of affinity proteomic and mass spectrometry, we are able to choose and drive and guide the researchers into the right technology between mass spectrometry and affinity proteomic when it comes to discovery projects, those that require a wide proteome type of analysis. But we also have some specific capacity to analyze, to perform proteogenomic or post-translational modification analysis, which are specific for identify or perform a better characterization of certain diseases. And one of our capacity is in particularly to measure proteins in biofluid, as I said, both with low, very sensitive immunoassays or microsampling, for which we are working with a system like dry blood spot that has been largely applied during COVID. And you can go to the next one.
No.
It's just like a final slide to say that we are 5 units, in particularly affinity proteomic, autoimmunity and serology profiling, glycoproteomic in Gothenburg, and global proteomic and proteogenomic will be involved in this type of call. And you can contact directly me or the platform director, Masoud Kamali Mogadam, and then we can direct you to the different units and our colleagues for this type of call. And I think if there is a discussion, I'm happy to discuss with any of you and give you more detailed information on what we can provide.
Yeah, thank you very much, Claudia. So this was the last presentation of this session that we have all together. If there are any general questions that anyone would like to ask before we open up the different breakout rooms with the platforms, feel free to ask them now by raising your hand or to write in the Q&A. Don't see any questions.